Okay, so we're going to go over the slides for the first presentation that is working according to the book, which is in accordance with chapter 1 of part 1. Okay, let's see here, let's get it started. And okay, so I'm not going to go over the first slides with all the warnings and notices. You can read them, and if you have any questions, just communicate them to me. So we're going to talk about the term that is called the genotype. A genotype is a sequence of DNA, a DNA sequence of a genetic makeup of a cell, and therefore of the whole organism or individual, if we're talking about people, which will determine a specific characteristic, which is called a phenotype of the cell, of the organism, of the individual. Okay, so basically when we say the word gene, we're talking about a genotype. A phenotype is the composite of an organism's observable characteristics or traits, which we have addressed in the first presentation, and we're going into greater resolutions of everything that we've discussed before and even more. So it can have something to do with morphology or development or biochemical aspects or physiological properties and aspects of existence. Phenology, behavior, or products of behavior. So anything that we can observe as a trait, ability, or skill. So we're encountering for the first time this slide that says see movie. All you need to do is take your cursor and click on the slide, and it will throw you to YouTube, and you'll see the movie there. Okay, and I hope you enjoy. Okay, so our genome, which is a term that you need to know, is actually a long sequence of nucleotide acids that include information. The information in this case is something that we called instructions, right? That can be interpreted and then translated, meaning that it can be transcribed into a more workable status that we called mRNA, and then translated into amino acids in order to create, in most of the examples, polypeptides. So the nucleic acids are building blocks, so to speak, of the genome. Okay, and the genome is actually all the information that we need in order to create an organism. It's important to understand that the genome itself is not active in the sense that it has to be processed in order for things to do, but it does determine hereditary features according to its nucleic acid sequence. Okay, and it includes all the information needed to produce the proteins needed for both building the organism and the organism's metabolism. Alright, once again, we have a movie that you can see. I hope you enjoy. And just a second, here we go. So when we look physically The genome is divided physically into nucleic acid molecules and functionally into genes. What does that mean? Every gene is going to influence a different kind of trait or function, but when you look at the genome, you can't immediately recognize the genes. It's just sequences of nucleic acids, which we are going to learn how to identify and understand which one is a gene, which one isn't, and so on and so forth. So every nucleic acid sequence molecule may contain many genes, okay, between 500 to 40,000 according to different organisms. So when we look at the genome, there's more than one gene there, okay. You have figure 1.1 on page 1 of the book. Please take a look at it, okay. And when you're referenced to a figure in the presentations, I won't read it again. It's just the first time, so please make sure that you look at it. So a gene is considered what we call a particular factor, meaning that it has a certain role to play, okay, and it passed unchanged from your parent to you, or from someone's parent to the next generation. So as of now, although it's going to change, one of the conceptions was that one gene represents one protein, and we'll discuss that and evolve the conversation around that in future chapters and videos. So we already talked about the allele in the introduction presentation, and it's actually an alternative form to a gene, okay? And you are advised to see a movie. So in diploid organisms, which include humans, there are two sets of chromosomes. The chromosomes carry our genes. We have one copy from the father, and we have one copy from the mother, right? The same goes for our genes. So the genetic location of a gene is its specific placement in the linear sequence of a gene in the chromosome. Okay? So if you have a sequence going from here to here, where on that sequence is located that gene, that is what we call a locus, right? So different alleles of a certain gene will also be found at this location or locus. Okay, so let's say you have alleles for the eye color, right, that we discussed before. They're all going to be found in that same locus, in that same location. Throughout evolution, the alleles for the same trait on the same chromosome tend to stay there, even when they're passed on to their next generation. And that phenomenon that they tend to be in the same areas in the same chromosome is what we call in genetics linkage. So now you're advised to see a movie. Once again, have fun. the microbial nature of our genome allows humanity to have more than a few progenies for a single set of parents, right? So we talked about that when we see the children to the parents, they're not exactly identical. So the process that is driving those differences is called recombination between genes, and we'll talk more and more about that, and we'll get a grasp of what that means. So we're chewing the materials bit by bit and using repetition to make sure that you got it all right. So the genome consists of sets of chromosomes for the entire organism. The genome determines the sequence of DNA for each chromosome. So we already know that in a chromosome we have sequences of DNA, and the genome is actually the whole picture of all the DNA needed for different chromosomes in order to produce an organism. Right, you have a movie, right? So right now we are saying that one DNA molecule equals one chromosome. So every gene codes for a specific protein. That is, of course, not the whole story. It means that we have information, we have instructions, and we have a product, and something has to happen in between to link the two of them. But what we're saying is that the gene is the information, includes the information that will end or result in the production of a specific protein. The gene is decoded into a unique amino acid sequence, the protein, right? Through utilization of a complex apparatus that we will talk about, but I'll just say that it's called the ribosome. And when we look at DNA molecules, we notice that they have two strands of nucleic acids, while its product after transcription, RNA, has only one strand. So DNA undergoes transcription that produces an RNA strand that has only one strand, while the DNA has two strands. The RNA strand is identical to one of the DNA strands from which it has been made. Okay? And now we're going to talk about protein coding genes. That means that that gene actually codes for a protein. And that means that it's going to have within the gene what we call protein coding DNA, producing a protein coding RNA, right? Once again, the RNA is transcribed from the DNA. The DNA has two strands, the RNA has one. So if the gene codes for a protein, it will have protein DNA, protein coding DNA, right? Which will produce a single strand RNA that codes for protein, and that will be decoded into a protein. So that last part shouldn't surprise you. Non-coding DNA will not result in a protein produced. Okay, so that's kind of logical, right? When you look at DNA, some of the DNA is called junk DNA, and we're going to discuss that a bit. So you have a movie, enjoy it. So when we look at the genome, at this level, we can divide it into non-coding DNA and coding DNA. Once more, I promise you we'd have a lot of repetition of the ideas. Non-coding DNA does not result in a protein. coding DNA results in a protein. We're going to stop here. More videos to follow.